we are group 6. We are presenting on catabolism of Haem into bilirubin. So on this topic, we are going to learn about how Haem is being catabolized into bilirubin. Yes, please. So we start by knowing some terms like the Haem, the bilirubin, and the catabolism. So Haem is the iron-containing part of hemoglobin that gives the blood its red color. So when you take the blood, the component that gives its red color is the Haem and allows it to carry oxygen. So the Haem, it first gives the color to the blood, that is the red color. And secondly, it also helps in carrying what we call oxygen. So the next term is bilirubin. We are saying that bilirubin is formed when bilirubin reductive converts the bilirubin, that's the green, into bilirubin yellow. So the bilirubin 2 is one other component, and what it does is that it is formed when the bilirubin, and the bilirubin is a reductive that converts the bilirubin, which is a green color, into a yellow color. Yes, and it is a lipid soluble in nature. So it is lipid in nature, and it is also hydrophobic. Yes. So we are saying that it does not dissolve in water easily. What happens is that the bilirubin does not dissolve in water easily. So in blood streams, it travels from the abdomen to reach the liver. So that is how come it travels. It travels from the bloodstream into the liver. Now, catabolism. Catabolism simply means that breaking down complex molecules into smaller ones. So when big molecules are being broken down into simpler ones, that is what catabolism. So we are saying that the Haem is in this case a bigger component. So the process that it goes through to be breaking down into the bilirubin in a smaller unit is through this process called what? Catabolism.
So we are going to talk about the site of heme catabolism. So one is the spleen, and the spleen is the primary graveyard of blood cells. So macrophages here involve centers. So the first site is the spleen. The spleen is the primary graveyard of red blood cells. So the macrophages here involve senescent red blood cells and then starts the heme breakdown. So the spleen, that's where the breakdown of the heme starts. And then the liver acts as a recycling and then a processing plant. So the puffer cells, which is the liver macrophages, continue the heme degradation, and then the hepatocytes, and then the bilirubin conjugation. So the bone marrow catabolizes heme from ineffective erythropoiesis, which is the immature red blood cells that don't make it to circulation, and the reticuloendothelial system, a system of macrophages in the spleen, liver, and then the bone marrow, and then the lymph node. And then we have the other tissues, which is the heme proteins like cytochromes or myoglobin, are also broken down into various organs, contributing to smaller share of bilirubin.
Yeah, I'm here to continue from where my sister left off. I'm going to talk about Haem oxygenase. This is the first and crucial step in the Haem catabolism. The moment Haem ring is cracked open, Haem oxygenase, which is an enzyme, should react. So what is Haem oxygenase? Haem oxygenase is the key enzyme responsible for the first and rate-limiting step in the breakdown of Haem into bilirubin. It converts Haem into bilirubin, the green pigment, and releases iron and carbon monoxide as byproducts. They are mainly macrophages of the spleen, liver, and bone marrow. There are two types of Haem oxygenase, the HO1 and the HO2.
So I'm talking about bilirubin reductase from Haem oxygenase. After the Haem oxygenase cracks open the Haem and produces bilirubin, the next step is handled by bilirubin reductase. And this is actually an enzyme, and it's a color changer. Bilirubin reductase is the enzyme that carries out the second step in Haem catabolism, like I said earlier, converting bilirubin, which is a green pigment, into bilirubin, which is a yellow pigment. It uses NADPH as a reducing agent. It produces unconjugated bilirubin, which is an indirect bilirubin, and it is lipid soluble. It occurs mainly in the macrophages after heme breakdown, and also in the liver and the spleen.
Good day. My name is Elana, and I'm here to continue with transport of bilirubin in the blood. Bilirubin is transported mainly in the blood by attaching itself to the albumin in its unconjugated form. Then it is carried to the liver where it is conjugated with glucuronic acid to become water soluble, and then it is excreted in the bowel. This transport system prevents bilirubin from accumulating in tissues, which otherwise can cause jaundice. Albumin binds to itself to albumin binds itself to bilirubin because bilirubin doesn't dissolve in water, so it can't really float in plasma. But albumin is abundant and soluble, so it acts like carrier protein, providing a safe way for bilirubin to travel in the bloodstream. Thank you.
Alright, continuing from where my colleague ended, is the hepatic uptake of bilirubin. So, with the hepatic uptake of bilirubin, it is the step where the bilirubin moves from the blood into the cells, the liver cells, that's the hepatocyte for processing. It arrives at the liver unconjugated and bound to the bilirubin. So it has three major steps that it goes through. The first step is the entry to the hepatocyte, that's the liver cells. So at the liver solids, the bilirubin dissociates from the albumin and it enters into the hepatocyte through the carrier-mediated transporters, mainly the OATP. So the second step is the intracellular binding. Here, the bilirubin binds to the cytosolic proteins, that's the ligand, and it is also called the GST, that's the glutathione S transferase. So it prevents bilirubin from diffusing back into the blood. So it stops bilirubin from diffusing again back into the blood. That is reversing the process all over again. And it's available for conjugation. So the third step is the preparation for conjugation. So now we are preparing it for what? For the conjugation. So the bilirubin is transported to the smooth endoplasmic reticulum, and it is now ready for conjugation with glucuronic acid.
Then I'm continuing from.
Continuing from where my colleagues ended, we have conjugation which occurs in the liver, mainly in the liver. So bilirubin in its natural form is insoluble in water. So it has to undergo conjugation where it becomes soluble in water and will be excreted in the intestines. So this reaction occurs in the hepatocyte. So mainly it binds with intracellular ligands where it is transported to the smooth endoplasmic reticulum. There it is conjugated with glucuronic acid where it occurs through the chemical reaction, giving what we call the bilirubin monoglucuronide as well as the bilirubin diglucuronide. They are the main forms of bilirubin in the blood. And then this reaction is facilitated by glucuronyl transferase. It is designed to facilitate this chemical reaction.
Intestinal fate of bilirubin. After conjugation in the liver, bilirubin is ready to be eliminated from the body. Conjugated or direct bilirubin is actively transported from the hepatocyte into the bowel canal and is transported by MRP2, which is the multi-drug resistance associated protein 2. It then flows through the bowel duct to the gallbladder and then into the intestine. In the small intestine, which is the colon, gut bacteria act on bilirubin. They convert it and deconjugate it into urobilinogen, which is a colorless compound.
possible pathways, which is the fecal route, the enterohepatic circulation, and renal excretion.